Good afternoon to everyone. I hope my screen is visible and the audio is fine, I guess. So, welcome you all to the NPTEL course on Environmental Biotechnology on the week 11 live interaction session for the course code NOC 25BT83. So, myself, Ajit Kumar, a PhD research scholar and PMR of fellowship in the Department of Bioscience and Bioengineering from IIT Goa. So this session we will be covering the week 1, week 11 content followed by the sample MCQs. Once again, I welcome you all to the course. Before moving on to the course, I will introduce myself briefly. Myself, Ajit Kumar, JDP Research Scholar, which is Joint Degree Program, PMR of Research Scholar, which is a research collaboration between IIT Guwahati and IIT BHU, Varanasi. So I work in a laboratory of biochemical and environmental engineering laboratory where our lab focuses on wastewater treatment in different aspects and in different industries. And my research inclines to the application of enzymes in the wastewater treatment, especially textile wastewater. So, moving on to my qualification, I have done BTech Biotechnology from Sastra University, Tamil Nadu, and MTech Industrial Biotechnology from NITK, Suratkal, Karnataka. And about my working experience, I worked in many pharma companies like Biocon and Enzyme Bioscience, where I worked on the pharma solutions. So this is all about my personal introduction. And without any delay, we will move on to the week 11 live interaction session. So as usual, this session will be of dividing into two offs. The first off will be we will be recapping the week 11 lectures and their important concepts and their applications, followed by the sample MCQs regarding the week 10 and week 11. So if you see in the last class, we have discussed briefly on the bioremediation, which is the application of environmental biotechnology, where we have seen important concept of point source and non-point source and what are all the examples and why it is necessary to know about the point and non-point sources. We have discussed briefly and followed by we also seen the nature of the pollutant and classification of pollutant, where we have discussed different classes of organic pollutant. And moreover, we have also seen some important terminologies in bioremediation, where we have discussed biotransformation, biodegradation, biomineralization, and activation. These are all some of the important concepts, along with their examples we have discussed. And also, we have seen factor affecting the bioremediation and what are all the fundamental prerequisites for microbial degradation, where we have discussed five principles on the fundamental prerequisites regarding the microbial degradation. And also, we have seen at last the availability of nutrients, where we have discussed the micronutrients and macronutrients, and we have seen what are all the essential and non-essential nutrients. And also, we have seen the other factors like temperature, pH, and absence of toxic or inhibitory substance, how it is affecting the biodegradation rate of progress. So these are all the some of the concepts that we have covered in the last class. And if you have missed out, you can visit the video of week 10 live interaction session. So the videos are recorded and it is published in YouTube. So you can visit at any time. So in this class, we will see mostly the application of bioremediation with different classes of pollutant and their common examples and what kind of metabolic pathway they are undergoing. So this is all about today's session. We will briefly move on to the application of the bioremediation, which is an important concept in environmental biotechnology. So if you see the common biotransformation mechanism, and we all know that biotransformation is nothing but using the microorganism or their derived products like enzymes, which helps in transforming, that is changing the pollutant from one changing the pollutant from one state to the another state. We call it as biotransformation. So, what are all the common biotransformation mechanism? We will see. The first, you have to see what are all the important classes of organic pollutant that prevail in the environment. They are mostly mineral oil constituent, which is mostly consists of aliphatic and aromatic compounds, and some halogenated products of petrochemicals. So these are all the important classes of organic pollutant. And if you see, in order to degrade these organic pollutant, mostly we prefer on the aerobic microorganism. And what do we mean by aerobic microorganism? This is nothing but the microorganism which uses oxygen as an final electron acceptor in order to degrade these classes of pollutant. And this is important for breaking down these kind of oil-based pollutant where oxygen will be playing an important role for high energy fuel for these microbes. So what happens under the aerobic mode of degradation? So microorganism will uses this oxygen and attacks the complex oil molecules. So eventually they are going to break down this complex aromatic ring and they are going to transform into a simpler product like CO2 or H2. We call this strategy as biomineralization. So with the help of this aerobic microorganism, we would be able to achieve this rapid and complete degradation through aerobic condition. So this is one of the most important concept of using the aerobic microorganism. And for example, we can take the deep water horizon oil spill, which occurred in the Gulf of Mexico, where um, tons of oils has been spilled accidentally into the ocean. And if you have gone through the earlier lecture, this case study we have discussed in briefly, where alkali forex microorganism, which has the capability to degrade this petroleum containing hydrocarbons. So what happens? This microorganism will consume these hydrocarbons and degrade them efficiently. So this is one of the example for biotransformation in case of oil spill. So if you see the aerobic microorganism, how does it degrade the organic pollutant? So what are all the essential characteristics of this aerobic microorganism for degrading this organic pollutant? There are four important steps that needs to be understood of how this aerobic microorganism helps in degrading this organic pollutant. The first important concept is they make contact with the pollutant. That is, we have to optimize the contact between the microbial cell versus pollutant because the microorganism, what happens, it can only degrade when the pollutant are in easy accessibility to the microorganism. Sometimes what happens, they produce a biosurfactant which helps in breaking down the oil into tiny droplets so that the pollution became accessible to the microorganism and then they degrade on this. So the first and most important point to remember is their accessibility or their contact between the microbial cells and the organic pollutant. So that what here you have to remember that the pollutant or the organic compound must be easily accessible to the organism that have the biodegrading capability. For example, if you see the hydrocarbons which are water insoluble, so in case of insoluble hydrocarbons, you what happens is the microorganism. tend to produce some surfactant which clump these hydrocarbons into one place, then biodegradation happens. So this is why the accessibility is the main factor for degrading the organic pollutant. And second important point is their oxidative attack. So what happens when the microorganism is accessible? Once the pollutant is accessible to the microorganism, the initial intracellular attack on the organic pollutant is their oxidative process. What happens here? The enzymes like oxygenase or peroxidase, which inserts or uses the oxygen pollutant molecule pollutant molecule, and they tend to activate the proper enzymatic key reactions, and they start the oxidation process. So the oxidation process is the key step, and it is an important role in degrading this organic pollutant. So once the pollutant is accessible to the organism, the next very step is their oxidative process. That is, they uses oxygen and they attack the organic molecule, and they break down these complex substances by breaking their aromatic rings, and eventually they will break down these compounds. And the third important step is their step by step degradation. So once the initial oxidative attack happens, it starts to degrade the pollutant step by step. That is, it converts the organic pollutant into intermediary metabolism. That is, if you assume a pollutant, a large complex molecule, first microorganism will utilize these pollutant and they degrade step by step into degradation intermediary metabolism using this degradation pathway to cut the complex molecule into smaller pieces. And these smaller pieces, what happens? It eventually enters into the central intermediary metabolism. That is, it enters into the glycolysis or TCA or ETC pathway in order to get energy from the substrate. So this is the third important step where step-by-step degradation occurs once oxidative process has initiated. And eventually the process will generate ATP as well. And the fourth important step is that building a new biomass. That is, once the microorganism degraded these large complex molecule into small molecules like acetyl CoA, succinate, or pyruvate, what happens? It starts to generate the biomass, that is, new cells or proteins or enzymes that are required for their cells. So it is nothing but we are converting the waste into wealth. So what happens here? These succinate and pyruvate, they enters into the TCA cycle and eventually generates the ATP as well. So they use these precursor metabolites for their growth because the pollutant don't usually get energy from the simple carbon. So the microorganism uses different strategy like glyconeogenesis. That is, they produce the sugar from the non-sugar sources like pollutant. So these are all the four important steps for the aerobic microorganism that is degrading the organic pollutant. So if you want to recall this, first the important step is the accessibility. That is the contact between the pollutant and the microorganism. Followed by once the accessibility has been achieved, then the initial step of oxidative attack will be initiated. Once oxidative attack has happened, it will undergo step by step conversion and they convert into intermediary precursors like acetyl, CoA, succinate, and pyruvate. And eventually, this helps in forming the biomass for their growth and for energy production. So this is these four important steps is essential characteristics of the aerobic microorganism that helps in degrading this organic pollutant. Then if you see the major organic pollutant in the contaminant sites are the first class is that aliphatic hydrocarbons. In aliphatic hydrocarbons, examples are alkane, alkene and alkyne, and cycloaliphatics and ethers. Whereas the second class, aromatic and polyaromatic compounds where benzene, toluene, ethylbenzene, xylene, etc. Phenols, naphthalines, etc. These are all like aromatic ring. That is, they contain the benzene ring in their structure. Then we have third class halogenated aliphatic, where examples are trichloroethylene, trichloro tetrachloroethylene, ethylene dibromide. So they contain halogenated atoms like chlorine, bromine in the aliphatic hydrocarbons. Whereas aromatics represents which is nothing but aromatic with halogenated atoms. Examples polychlorinated biphenyls, pentachlorophenyl, phenol, dichlorobenzene, chlorophenoxylates. So these are all some of the examples for halogenate aromatic. Then nitroaromatic, which is nothing but aromatic ring with nitrile group. For example, nitro phenol group with nitro functional group, which we call it as nitrophenol, nitrobenzene, and nitrotoluene. These are all some of the examples which contain nitro as their functional group. And last is the pesticides or herbicides, which is also an organic pollutant. Example, organophosphorus, organochlorine, or phenolic compounds. So these are all the different organic pollutants that exist in the contaminant sites. So here we will see the basic metabolic pathway of aliphatic and aromatic compounds. So if you see the degradative pathway of aliphatic breakdown, and as we have seen in the previous slide, some of the examples are N-alkane, alkene and alkyne. These are all aliphatic organic pollutant that exist in the contaminant. So what happens and what is this? These are all tough alkyne and alkene, which is composed of long hydrocarbon chains, which has the application in many fuels and oil industries. So, in order to degrade these organic pollutants, there are three different ways where these pollutants are degraded. The first one is their aerobic mode of degradation. And what happens here? The in aerobic mode, the microorganism will utilize alkane, alkene, or alkyne by secreting the enzyme called oxygenase. So these oxygenase enzyme attacks the alkene structure and they convert into alcohol through oxygenation reaction. Once it has formed alcohol, it converts into aldehyde by secreting alcohol dehydrogenase enzyme, and then alcohol aldehyde will be converting into fatty acid through aldehyde dehydrogenase enzyme, where dehydrogenase means removal of hydrogen atom from their structure. So eventually what happens, the fatty acid will be formed and these fatty acid will be converting into acetyl CoA through beta oxidation, that is cleaving the chain of fatty acid at the beta position. And this acetyl CoA, what happens? Once it is formed, it will enters into the central metabolic pathway where it will enter into the TCA cycle and eventually ATP is generated. So this is how the complex alkane, alkene or alkyne structures are converted into simpler precursors, which again it will be utilized by the microbes for generation of ATP. So this is the aerobic mode of degradation. But what happens if anaerobic organisms are used? So the first step alone it will change where in anaerobic mode of degradation, instead of oxygenase enzyme, they undergo hydration reaction, which is nothing but they add water and they start the reaction by breaking down their structure into alcohol. Once it has converted into alcohol, the rest all are same where alcohol will be converting into aldehyde through aldehyde dehydrogenase enzyme, and aldehyde will be converting into fatty acid by aldehyde dehydrogenase enzyme, and fatty acid will be eventually converting into acetyl CoA, where it will enters into the TCA cycle for energy and biomass synthesis. So the only difference is their initial step, where if it undergoes oxygenation, it is an aerobic mode of degradation. If it is undergone dehydration step, it is it implies that it has undergone anaerobic mode of degradation. It depends upon the species to species, and in anaerobic itself, if carboxylation of alkane structure occurs, that is, if we add the CO2, it eventually converts them into directly fatty acid, and these fatty acid convert into acetyl CoA through beta oxidation of fatty acid, and eventually it enters into the uh, TCA cycle. So this is how the three different degradation pathway generally microorganism adapts based on their environmental condition. And first is the oxygenation mode, second is the hydration reaction will be occurring, third is carboxylation reaction. And why it is important to understand these aliphatic breakdown? Because, for example, in case of oil spill, aerobic microorganism will rapidly degrade these hydrocarbons at the surface. And in oxygen depleted zone, what happens? These microorganism will undergo anaerobic mode of degradation. So here nothing is wasted; it all converts waste into wealth. So depending upon the condition, these microorganisms acts and produce appropriate enzymes in order to completely degrade these aliphatic compounds. So this is regarding the aliphatic breakdown of degradative pathway. And if you see the next case, which is degradation of aromatic compounds. And what happens in the degradation of aromatic compounds? And we know that these aromatic compounds are they are stable ring structure and they are tough to degrade. And examples of aromatic compounds are, as we have seen earlier, it is benzene, toluene, xylene, etc. So how does it degrade? First, in case of aerobic biodegradation of this class of aromatic compound is common. And what happens? The first step is to convert into the key intermediary product like catechol. And how it is converting them into catechol? First, what happens? We will take one such example, which is benzene. The benzyl will undergo oxidation reaction where hydroxylation happens, which is catalyzed by the deoxygenase enzyme. So first the benzene will be oxidized by dioxygenation enzyme, which forms diol. And this diol will be converting into catechol through synthesis of dehydrogenase enzyme. So this is how the aromatic compound like benzene converts the catechol. Once catechol is formed, it undergoes various steps in the metabolic pathway, which unlocks the aromatic ring through various enzymatic cuts. So, this is the main key intermediate that we found in most of the aromatic compound. If you take toluene, xylene, eventually it will be converting into an intermediary product called catechol. And catechol will be again sent into the central intermediary pathway to complete the reaction. So here what happens, we will see in this diagrammatic representation also, but here you have to remember that all classes of aromatic compound, first they undergo hydroxylation reaction in order to convert them into diol. And through dehydrogenation, it will convert the diol to catechol reaction. So this is the common pathway that is observed among various classes of aromatic hydrocarbons. And if you see the degradative pathway, as I said earlier, the aromatic compounds like either it is a benzene, phenol, toluene, aniline, or anthracene or naphthalene, through hydroxylation and dehydrogenation reaction, it will be converting into catechol. What happens here? The intermediary product of diol will be formed, and this diol will be converting into catechol through dehydrogenation enzymes. Once the catechol is formed, this is the common step that occurs across different aromatic compounds. The first, if the catechol structure is cleaved at the O position, then these degradative pathway will be occurring. First, it will be converting into cis muconate, followed by muconolactone, which again it will be converting into 3-oxoadipate enolactone, and then 3-oxoadipate lactone. Then through aerobic breakdown, it will be converting into succinate and acetyl CoA. And we all know that this intermediary product again it will enter into the TCA cycle in order to produce the ATP and biosynthesis of cells. So this is how the complex aromatic compound converting into a simple precursors. And if the cleavage occurs at the meta position, what happens? It will undergo these cycles, and in each step, different enzymes will be produced, and it helps in catalyzing the next reaction. And if the cleavage occurs at the meta position, what happens? It will convert into hydroxymuconic semialdehyde. It will convert into oxopentafluoronate, which will be converting into 4-hydroxy 2-oxovariate, and then through aerobic breakdown, it will eventually convert into acetaldehyde and pyruvate. And we all know that pyruvate is the product of glycolysis cycle, and eventually it will be going into TCA cycle and then ETC for production of ATP and biosynthesis of cells. So this is how the various classes of aromatic compounds converting into a key intermediary product called catechol, which again converts into a simpler product through various enzyme degradation mechanism. So why this is important to study these degradation pathway of aromatic compound? Because in polluted river or soil near the petrol station or industrial wastewater, we observe these kind of aromatic compounds normally. So the microbes rely on this catechol pathway to break down these kind of hazardous compounds like benzene, toluene, aniline, anthracene, and naphthalene compounds. Because if these compounds present in a very less quantity, it is very dangerous for the environment. So this is why it is very important to understand this degradation pathway, and this helps us to select the microorganism effectively. So till now we have seen two important degradation pathway. One is regarding with the aliphatic, where we have seen alkene and alkyne compounds, and the next is we have seen the aromatic compound. And the next topic is how and what are all the modes of biodegradation pathway that microorganism undergo. Because usually what happens, the microorganism uses their own clever strategies to deal with different kind of pollutant. And these strategies are mainly they use these two kinds of strategy. One is growth associated strategy and next is co-metabolism strategy. And what is growth associated strategy? It is nothing but here the microorganism will consume the pollutant and consume and assume as their sole source of carbon and energy. So it becomes the main food source, that is main carbon source for the microorganism. So it uses these pollutant directly for their energy as well as to make more microbes, that is to synthesis more biomass. So what happens here? The pollutant will act as an food source and it helps in attaining their own energy. So if you see here, the methane is converting into uh, intermediary product through different reactions where the methane is directly assumed as an source of food for the microbes. This is the growth associated strategy. Whereas in co-metabolism, what happens? As we know that some chemicals or some organic pollutant are stubborn and these microorganisms cannot utilize directly and assume these pollutant as their own food. At that case, what happens here is the microorganism will consume a simpler source of pollutant. For example, in an environment where methane is also present and the TCA, trichloroethylene, is also present. So at that case, what happens is the microorganism first consume the CH4, methane as a substrate, and they convert into CH as their first reaction. So in this case, the microorganism will secrete methane monooxygenase enzyme to an higher level. And this enzyme, what happens? This helps in converting this TCA into TCA epoxide, which eventually break down to form a CO2 or glycoside. So this is the co-metabolism strategy where microorganism cannot directly assume the TCA as an one of their food source. In this case, they will always assume the simpler carbon source to assimilate, and they secrete some kind of enzymes or product which help indirectly degrade the pollutant. So this is the co-metabolism strategy where metabolism of organic pollutant in presence of another growth substrate. Here the another growth substrate refers to the CH4, which is primarily used as a carbon and energy source. So this is the two clever strategy that microorganism uses to deal with the stubborn organic pollutant. And one is, what is it? First, the pollutant itself act as an carbon source. In second case, what happens? The pollutant that needs to be cleaned, they will assimilate simpler sources, and using their metabolites or products, they tend to clean up the pollutant. So this is the two clever strategy that microorganism follows. And and till now we have seen different degradative pathway. And if you see in each pathway, there are different enzymes that play an important role in order to catalyze the reactions, right? So if you see the different classes of enzymes, there are six major classes of enzymes that are involved in this biotransformation or biodegradation reaction. And the first enzyme is oxidoreductase, which is nothing but it helps in catalyzing both oxidation reaction and reduction reaction. And what is oxidation reaction? It is nothing but oxidation means it is a loss of electron. That is, they take away the electron from them. Whereas reduction means they add electron, they gain electron from the reactions. So these enzymes will catalyze both oxidation and reduction reaction. And if you see among the classes of enzymes, oxidoreductase is the largest class of enzymes, and they are most commonly used enzyme in case of biodegradation. And what happens here and why this enzyme is chosen for biodegradation? Because of their mineralization capacity. So mineralization of this organic compound often involves the oxidation, and they build their own cellular product. And if you see the common oxidoreductase enzyme, the one is dehydrogenase enzymes. The name itself telling us dehydro, that is removal of hydrogen atom from the structure. This is one class of subclass of oxidoreductase enzyme, and the another subclass is their oxygenase, where it helps to add the oxygen as OH functional group to the hydrocarbons to facilitate further metabolism. So dehydrogenase means removing the hydrogen atom, whereas oxygenase means adding the oxygen in the form of hydroxyl functional group. So if you see many of these oxygenase enzyme, they attack the aromatic hydrocarbons. And we have also seen in the degradative pathway of aromatic compound where oxygenase play an important role in degrading the metabolism. For example, if you see the toluene deoxygenase enzyme, this enzyme particularly they are capable of degrading more than 100 different compounds, which includes nitrobenzene, chlorobenzene, and TCl. So this kind of enzyme is very famous and they are effective in degrading many kind of pollutant. So this is comes under the first class of enzyme, which is oxidoreductase, where this enzyme help in catalyzing both oxidation and reduction reaction. And this is the one of the largest class of enzyme. So if you see the second class of enzyme, which is hydrolase, it acts as a molecular scissor. What happens? It uses the water to perform the hydrolysis to cut the chemical bond of CO, CN and CC bond. And where do they work? They especially break the bond that are found in the amide, ester, phosphate ester, monohaloalkanes and epoxides. So these hydrolase they attack on the CO, CN, CC bond that are found in these structures. And what are all the common types that are under hydrolase enzyme? One is esterase, the term itself suggesting us that it breaks down the ester bond that are formed between the organic compounds by adding the water. And depolymerase means removing the polymers. That is, they hydrolyze. They cleave the polymers into monomers. And third class is dehalogenase, which removes the halogen atom from the compound and they replace them with the OH groups. So hydrolase is nothing but it acts as a molecular scissor to cut the chemical bonds using the water molecules. And this is the second largest class of enzymes after oxidoreductase. And third class of enzyme, which is nothing but lyase. What does it does? It does remove the functional group molecules without using the water. That is why it catalyzes either non-hydrolytic removal of functional group from the substrate with resulting formation of double bond or reverse reaction. So what happens here is the key players are dehydroxylation, carboxylation reaction. That is, they remove the COOH carboxyl group from the compound. And second step is hydration, that is they add the water which helps in breaking down these alkanes or alkanes aromatic compound which converts these hydrocarbons into simpler compounds that microorganism can metabolize. So this acts as an enzyme which helps in catalyzing or removing the functional groups from the molecules without using the water. So the next class of enzyme is the transferase, where these enzyme helps in transfer the functional group to the organic compound. What kind of functional group? It can be either thiol group like SH or hydroxyl group like OH. And why it is so? It is mainly essential for cell synthesis and sometimes for detoxification. For example, GST, which is glutathione S transferase. What happens here? It is essential for transferring the thiol group, that is SH, to the chlorinated compound with dechlorination. So this helps in reducing their transferring the SH group to the chlorinated compound. But some reactions may become trouble by using this transferase enzyme. For example, if there is an heavy metal and if there is any methylation occurs in this heavy metal, like you can take any mercury, arsenic, or strontium, what happens? It will transfer the methyl group to the uh, metals and they form methyl heavy metals, that is CH3 heavy metals. So in this case, what happens? Instead of detoxifying it, they try to increase their toxicity and their bioaccumulation potential. So it makes more toxic and increases their bioaccumulation capability. So there is a two-way purpose. One, it can be very useful, but at the same time, in case of heavy metals, it has shown more toxic effect and their bioaccumulation potential. So this is the fourth class of enzyme where transferase helps in transferring some of the important functional groups to the organic compound, like SH, thiol groups or hydroxyl groups. Hydroxyl groups. So this is the fourth class of enzyme, which is transferase. And the fifth class of enzyme is nothing but trans isomerase. Isomerase is nothing but it acts as a molecular rearranger. So it rearranges the structure of the organic molecule. So here they don't add or they remove anything. Instead, they rearrange the molecules internally to create isomers. So what happens here? They rearrange the structure of the organic molecule and they form new structure of the same molecular size. For example, this racemase enzyme which helps in catalyzing L2D or D2L amino acid interconversion. Whereas the sixth class and the last class of enzyme is ligase, where it acts as an building bridge. That is, they used to form the covalent bond, which is similar to the isomerase, where it helps in arranging the structures, right? So this ligase helps in catalyzing and forming the covalent bond. So these bonds prepare the molecule for further metabolism and it acts as an builders for enzyme world. So one such example is CoA ligase, which adds CoA to the fatty acid during the beta oxidation of fatty acid, where it convert into acetyl CoA. So these are all the six major class of enzymes that are commonly used in the biotransformation or biodegradation reaction. And the first is the largest class of enzyme, which is oxidoreductase. What happens here? They catalyze both oxidation and reduction reaction. Whereas in hydrolase, they used to hydrolyze by adding the water molecule and they cleave the CO, C and CC bonds. Whereas in the third class, lyase, they catalyze either the non-hydrolytic removal of functional group from the substrate. Whereas in transferase, they transfer the functional group to the organic compounds. What kind of functional group? Either OH or SH group. And the fifth class of enzymes or isomerase, it acts as a molecular rearrangement. That is, it uh, rearrange the intramolecular structure of organic molecule. And the sixth one, last is ligase, which helps to form the covalent bond, acts as an um, builder of enzyme world. That is, it used to catalyze the covalent bond formation. So these are all the important classes of enzymes. And if you see what are all the common biotransformation or biodegradation reaction, in each step, it has been categorized into four reactions. So these are all the four different reactions that normally seen in the pathway of the biotransformation or biodegradation. And the first one is oxidative transformation. What happens here? In oxidative transformation, it adds the oxygen or remove the electron from the organic compound and makes the chemical more reactive and it becomes easier for their breakdown. For example, in case of alkane, they use hydroxylation reaction in order to break down this reaction. Whereas in the case of atrazine, dealkylation will be taking place. In case of TC, trichloroethylene, epooxidation will be taking place. And in case of aromatic hydrocarbons, oxidative ring cleavage will be occurring through oxidative transformation. And the second most reactions are reductive transformation, where in the absence of oxygen, the reduction reaction occurs. That is, they remove the oxygen molecule and make the toxic compound safer limit. And one such reactions is reductive halogenation, which is seen in the halogenated compounds. And in case of nitroaromatics, RDX or TNT, we observe reduction of nitro groups. The first reaction will be removing this nitro functional groups from the structures. So these kind of reactions comes under the reductive transformation. And third class of reactions is hydrolytic or other biotransformation, which is not involving in the redox process. That is, it acts as a molecular scissor. For such reactions are hydrolysis, hydrolytic dehalogenation, and dehydrogenation. And last and most important reactions are synthetic reaction. What happens here? They act as a builder. So they don't break up any things, they build new things from the organic molecules that undergone degradation. So if it adds the carboxylation group like COH, we call it as carboxylation reaction. If it adds methyl group, we call it as CH3. So these are all the four strategy, which is oxidative transformation, reductive transformation, hydrolytic and other biotransformation, and the last is synthetic reaction. So these are all the four different strategy which shows us microorganism acts as an environmental guardians, which helps in cleaning up the pollutant that humans created and released into the environment. So these are all the four important reactions that occurs during the biotransformation or biodegradation reaction. Then if you see the remedial goals of this environmental pollution, these microbes acts as an environmental doctors. And how does it achieve these goals? The first is through precipitation. That is, the microorganism first it triggers the chemical reaction, which converts the undissolved metals into solid particles. Once it became precipitated, the metal falls out from the water and locked in the soil. So by which we will easily separate out some of the inorganic contaminant and we can accumulate this pollutant. And second is we will be concentrating them. By concentrating them, we can achieve the reduction in the volume of the contaminated matrix. That is, we are gathering all the dirt into one spot. That is, the microorganism will concentrate the toxic metal into smaller volume. So this reduces the volume of the contaminated area, make it easier for us to handle, treat and safely dispose these hazardous contaminants. And third is we will compartmentalization of these metals. That is the contaminants can be trapped either in the deep underground or inside the mineral lattice. So where they remain quarantined from the outside world, from the plants, animals, and humans. So these are all some of the remedial goals that can be achieved. First is the precipitating the inorganic contaminants, or we can concentrate and reduce the volume in case of our contaminated matrix, and we can compartmentalize and quarantine them by not reaching into the public. So by this, till this, we have seen the organic pollutant, and in this, we have seen different classes of organic pollutant and their degradation pathway. Like we have seen what if in the case of aromatic compounds, what if in the case of aliphatic compounds, and we have seen a different strategy how the microorganism adapt themselves to degrade this organic compound. One, we have seen the growth associated and co-metabolism strategy. So by this, we completed the organic compound degradation. Now we will move on to the problem of heavy metals. If you see the problem of the heavy metals, and as we already know, these heavy metals, they usually exist naturally in the earth crust, and their amount is also very very less in the ppm. Ppm means you can imagine 1 mg of heavy metal in 1 liter of media, but this is sufficient to affect the living organism. So normally if you see these heavy metals, they seen in the very tiny concentration or trace concentration like ppm, but at this trace level, some heavy metals, some metals can act as an vitamins or supporting enzymes and helps the plants, animals, or even us to stay healthy. These metals, but there are some heavy metals they start to accumulate in the food chain, and these organism absorbs just little, and this concentration grows up very stronger as the predator eat the prey. So there are some useful metals and hazardous metals too. So the disposal of these heavy metals from metal excavation and processing have concentrated these metals to the dangerous level. That is, the heavy metals like arsenic, cadmium, these are all, even in the very less quantity, they became very hazardous, not only to the wildlife, but also to the people and to the environment. So scientists are now taking many efforts to remove these heavy metals from the soil. And we already know the microorganisms have coexisted. The microorganisms have been coexisted with the metals since the evolution, even before the humans has formed. So, different microorganism has been evolved in the evolution to withstand wide range of divalent or transition metals, and some metals are also acting as an enzyme active sites. So by this we understood the problem of heavy metal, and these heavy metals are always seen as two-faced coins. That is, they can help as well as it can destroy the environment. And if you see, some metals are the part of their catalytic structure of the protein. So if you take some of the important proteins or biomolecules, what happens here is some metals like calcium or magnesium, they help in they participate in the normal cell metabolism. They act as a coenzyme or cofactor to catalyze the reactions. But these metals needs to be present in the very minute quantity for the normal cell metabolism. And their intake is also subjected to the requirement to the limited level. But some metals are there, they don't have any biological relevant function. For example, the metals like mercury or cadmium, they don't have any role in the biological function. Even a very minute quantity in the ppm level will cause a drastic toxic to the humans. They cause damage. Why? Mainly due to their presence of the sulfonyl groups of proteins. They block these and inactivate the particular protein even in the presence of few ppm. So this is the two side of the heavy metals. And from the physiological point of view, again the heavy metals has been categorized into three main categories. One is essential and basically non-toxic. You can think these kind of metals as friends. That is, they are essential helpers. So some metals they are, yes, they are needed for essential of life. For example, if you see the calcium and magnesium, these are all acts as a vitamins for the microbes. These are all micronutrients that are essential for the microorganism for their growth. For example, in calcium, it helps in stabilizing the cell wall of the microorganism, whereas magnesium it stabilizes the ribosome and cell protein factories. So there are some metals which can help, and they are essential and they are basically non-toxic to the some extent. And there are second class of metals where it is essential but harmful at the very high concentration. This means if these metals are, for example, iron, manganese, zinc, copper, cobalt, nickel, and molybdenum, these are all essential metals, but if they are present at very high concentration, it is obviously going to be hazardous for the environment. So, what happens? These metals are essential, but they are dangerous at the very high amount. So, they are required in the tiny quantity because they act at the active site and they catalyze and demineralize these chemical reactions. So balancing is very important in case of second category. Whereas the third category, which is toxic and hazardous, And some metals are very dangerous. For example, mercury and cadmium, as we have seen earlier, they have no biological relevant function. What happens? They tend to attack the protein that keeps the cell alive. So they attack the sulfonyl group of protein so that they block the usual function of these enzymes or proteins. So these are all the three main category of metals. The first is essential, but basically they are non-toxic. And second class is they are essential but hazardous at the very high concentration. And third category is they are very toxic as we seen in the case of mercury and cadmium. Even a very minute quantity can cause an hazardous effect to the environment. And if you see the heavy metal contamination, the microbes acts as a survivor in this world. That is, the microbes they evolve and they survived and adapted various tricks to develop the resistant against this heavy metal. So they develop various interaction mechanism with the metals. What kind of interaction? Some microorganism can pump out the heavy metal from their cells, or some can bind to them onto their surface, or some can reduce their toxicity level. As we seen in the case of uranium. What happens to the uranium? It converts uranium 6 plus to 3 plus, which is less toxic to the environment. And and these microbes, they are dealing with the metals long before the human discovered these metals. So generally, the emergence of metal tolerance has been seen in various microorganisms. But due to lot of human activities that has been observed in the mining, industrial waste or improper disposal of these batteries, what happens? They releases lot of heavy metals into the environment, which created metal loaded environment. So what happens here during the evolution, some microorganisms adapt and some died at the evolution. And those are adapted, they created new tolerance mechanism or sometimes they pass on their resistant genes to the next generation and they survive for generation after generation. So by this we understood that there is microorganisms that have evolved during the evolution in order to tolerate these heavy metals. And if you see the heavy metals interaction with the microbes, and we understood from the earlier lectures that microorganisms are powerful, but they are not magicians or they are not alchemists, right? They cannot destroy the metals, but they can transform or change the metals from one form to the other form in different strategies. I will be discussing the strategies. So, what happens? The first, what can they do? They can alter or change the properties of their heavy metals. That is, they can reduce their metal form by changing their oxidation state. Or otherwise, they can precipitate these metals that is soluble to insoluble form. Or they can tightly bind these heavy metals to their surface. And next is these microorganisms we call as an environmental engineers because they transform these heavy metals to different oxidation state. That is, for example, the uranium 6 plus to 4 plus, as we have discussed, which is 4 plus, is makes them less toxic and they are less mobile and less dangerous. So these microbes acts as a natural water filter. So these are all different strategies that they undergo. And if you see in the right hand side, this is the microbial cell, and these are different strategy that they undergo once they detect the heavy metals in the environment. The first strategy is their bioabsorption. What happens here? These heavy metals, due to the presence of their functional groups, that are present in the microbial cell. Mostly phosphate groups will be present on their membrane. So due to their ionic interaction between them, they adsorb onto the surface of the membrane of microbial cells. We call this strategy as bioabsorption. And the next strategy is bioaccumulation. The term itself explaining the accumulation of metals inside their cell. What happens? These metals will be accumulated into their cells indefinitely until a threshold level. So here accumulation of metals will be occurring inside the cell. Next is microbially enhanced chemisorption of metals. That is, using the microbes, they used to absorb these metals onto their surface. And the next strategy is to biomineralize them. What happens here? They tend to mineralize the metals by forming a complex between them. For example, the H2S plus M2 2 plus gives they form complex with the sulfonyl group that are present in the hydrogen sulfide. So it forms an absolute non-toxic form. Then some using the chelating agents, we will precipitate these heavy metals. And the next strategy will be biotransformation, which is also called as bioreduction. We will be reducing the toxic level of the heavy metal. That is, the metals will be oxidized and converting into non-toxic form by utilizing the electrons from the microbial cell. And now these heavy metals, these some of the metals have been used for leaching also. That is, bioleaching. You have been seen in the case of copper to extract some mineral ores from the environment. Like in order to extract the copper or gold, you can use an appropriate enzymes like ferredoxin enzymes are there, which helps to extract these copper ores or gold from the environment. So we call this strategy as bioleaching. So by this, we understood that the microorganism interact with the heavy metals in different ways. One is they absorb onto the surface, or they accumulate onto their membrane, or they try to mineralize them, or they try to reduce their form, or they try to leach from the surface. So these are all the different interaction of metal microbe that is normally seen in the environment for bioremediation. And if you see the first case, which is bioabsorption, which is nothing but a metabolism independent physiochemical uptake of metal ions. So if you see the bioabsorption, what happens? These heavy metals through electrostatic interaction with the cell wall, they bind to this cell membrane. We call this as chemisorption to ligands in case of cell wall. So what happens? It's nothing but these heavy metals will be absorbed onto the cell membrane where various kinds of interaction. It can be hydroxyl interaction, amine interaction, carboxyl or phosphoryl groups depending upon their electrostatic interaction. They bind. So it is normally a metabolism independent absorption of heavy metals. What happens here? Normally we know the cell surface will be carrying a net negative charges on their surface. So positively charged cationic metals will be easily absorbed onto the surface due to the electrostatic interaction. And why there is a negative charge at the surface? Because of the presence of different groups like carboxyl, amine groups, hydroxyl, phosphate or sulfonyl groups. So these are all the different groups that are present, which helps us to absorb an appreciable quantity of positively charged cationic metals. Then how it differs from the bioaccumulation? In bioaccumulation, what happens? The penetration will be occurring. Penetration of any heavy metals will be observed by increasing the membrane permeability. So what happens here? The uranium will be entering into the or inside the cells through membrane transporters. What happens first? It will interact with the negatively charged functional groups that are present on the surface of the cells. Once it observes, uptake of these heavy metals will be the taking place. So first adsorption followed by the bioaccumulation. So this is the major difference between the bioabsorption and bioaccumulation, where bioabsorption means it's a surface phenomena where heavy metals will be attaching onto the surface only. Whereas in bioaccumulation, the heavy metals will be accumulating onto their cells. And what are the advantage? So bioabsorption methods seem to be effective even at the very low concentration. And the main advantage is we can use low cost waste materials like biomass sources. And sometimes dead biomass is also used for better adsorption capacity as the effect of metal toxicity is not as important. And this adsorption has very rapid kinetics and higher adsorption capacity. So a scientist or researchers should use an appropriate adsorption material in order to achieve higher adsorption capacity of these heavy metals. Then it is easier to remove the bound metals from the cell surface to regenerate the bioabsorbent. So the main thing is the high specificity of this bioabsorbent and their adsorption capacity. So these are all the two important factors that you need to take care while using the strategy of bioabsorption. And what are all the disadvantage of this bioabsorption? The first is the desorption will be as rapid as the adsorption. So you need to strategize the technique which can minimize the desorption capacity unless until you provide a strong functional group. So to attain this, you can modify the functional group by adding different reagents, so which helps to solve this problem. And these sorbed materials could be re-released into the solution when cells dies and decompose. Other cations compete. There are other cations also will be present. Since it's an electrostatic interaction, there might be an interaction of calcium or magnesium also, which is essential. But in this case, what happens? The heavy metal absorption capacity will be minimized. And the cell surface can also quickly become saturated, preventing further bioabsorption. So these are all some of the problems associated with this bioabsorption, but it can be used for treating the contaminant effluent in a pump and treat strategy, but it is not adequate for long-term solution. So it can be used for short-term solution. For long term, you have to go for biomineralization or biodegradation, by which a complete transformation of the heavy metals can be achieved. And the next strategy is metal precipitation. Here, what happens? The bacteria tend to precipitate the heavy metals or the radionucleotides by reducing the element to a lower redox state, producing less bioactive metal species. And what happens here? They tend to reduce the toxic metal through inorganic ligand produced. So in metal precipitation, there are two strategies. One is bioreduction, where you can reduce these elements to lower redox state. That is, the UO2 2 plus is converting into 1 plus. Uranium is converting into uranate. Or you can convert UO2 to U4 plus. So here what scientists will be trying to change their oxidation state to the lower oxidation state, which producing less bioactive metal species. Whereas in biomineralization, they tend to first gather all the uranium and they try to change the form. So here you could see it is a process by which metals first precipitate with microbially generated ligands. You could see the sulfides and phosphates or carbonates or hydroxates in response to the alkaline condition at the center. So first what happens by binding with the phosphate, they bind to the surface of the structure. So for example, if you see the Citrobacter species, the organic phosphate helps in precipitating this method by forming high phosphoric acid along with metal. So metal phosphate precipitation will be forming with the help of the phosphatase enzyme. So these are all the different strategies. One is bioreduction, biomineralization, bioaccumulation, bioabsorption. So these are all different type of interaction of metal radioactive nucleides interaction with the microorganism. So if you see the heavy metal treatment, as we have discussed a different strategy, and if you see mainly in the cleaning up of metals or radionucleated pollution from the soil, water, or any industrial wastewater, they use these kind of strategies like biomineralization, bioreduction, bioleaching, etc. So if you see these heavy metals, they normally coexist in the waste with other kind of pollutant such as organic pollutant. So if you see the real-time wastewater, the presence of heavy metal is inevitable. So you have to find a strategy so that it can detoxify all these organic contaminant as well as the heavy metals. So what kind of approaches you can follow? The first is you have to find a multi-talented microbes. That is using single microbes, we cannot achieve the oxidation of both heavy metals and the organic pollutants. So you have to use the microbes with multiple detoxifying functions. So in this case, you can use the microbial consortia, which is developed to oxidize the phenol while simultaneously reduce the chromium 6 toxic heavy metal. So you have to find an microbial consortia. There are different microbial consortia that has been developed not only degrade the organic pollutant but also reduce the heavy metals. And also you can use super strains, that is genetically engineered strains for dual toxification. That is, you can engineer these strains that can detoxify both which is an heavy metal, and toluene, which is an aromatic compound, which is normally you will be seeing in the radioactive waste. So you can find an engineered strain which can handle both mercury and toluene and degrade them to the safer level. And if you see, one such example is that Pyrobaculum islandicum, which is an hyper thermophilic bacteria, which is a nature's extreme specialist, which has a multiple metal reduction. That is, it simultaneously can reduce different metals like uranium, chromium, cobalt, etc. by using hydrogen as the electron donor. And moreover, this enzyme catalyzes the metal reduction at around 100 degrees Celsius also, as it is an hyperthermophilic bacteria. So there are some specialized microorganisms that exist in the environment which can be utilized for degrading multiple heavy metals, or you can handle different kind of organic pollutant along with the heavy metals. So by this, we could understand that no single treatment is enough for degrading the real-time wastewater treatment. So first you can use a consortia of microorganism, or you can go for genetically super strains that can do multitask. Sometimes we can change or genetically modify them using the advanced techniques like CRISPRs also. So in short, I want to convey that the microbes, they are not only survivors or chemists or engineers, uh, they offer smarter, greener way to tackle these heavy metals that were made by the humans. So finding an appropriate microorganism is more crucial for handling these kind of wastewater. So these are all the topics that I want to discuss this week. So we have seen different types of pollutant, like we have seen pollutant, aromatic pollutant, then we have seen aliphatic compounds, and we have seen the problem of heavy metals. What are the different strategies that microorganism follows to degrade or to handle these heavy metals, and what are the possible strategies or the techniques that you can approach for remediation of these heavy metals. So these are all the content for today. So if you have any doubts, you can post your queries in the chat box. And if you do not have any queries, we can move on to the MCQs. And you are aware that MCQ will be a discussion type of thing. So I will read out the question. You can post your answers in the chat box, or you can unmute your mic and you can say your answer. Okay. So I think we can move on to the MCQs now. So the first MCQ is, which of the following contaminants act as an electron donor? So option A, uranium oxide, option B, SO4, option C, chlorinated hydrocarbons, option D, benzene, option E, TNT, trinitrotoluene. So among these, which contaminant acts as an electron donor? So you can read out the question and post your answers in the chat box. So which of these following contaminant act as an electron donor? Your answers are, which of these following contaminants acts as an electron donor? Option A. Okay. So let's see the answer. No, it is option D, benzene. We will see why benzene. First, what happens? What is electron donor? It is nothing but it can donate electron, any molecule or ion that can donate the electron to the another molecule or ion in a chemical reaction, right? So what about the benzene ring structure? It has a stable ring structure with delocalized pi electrons, right? If you see the benzene structure, it is an aromatic compound which makes it capable of donating the electrons in certain circumstances. So it is a reduced aromatic hydrocarbon that can be oxidized by some microorganism. So it donates the electron by oxidizing these substrate by some of the microbes. So the correct answer is benzene. And why it is so? And why not the other options? If you see the uranium oxide, mostly it acts as an electron acceptor. Why it is so? Because due to their ability to gain the electrons and they form higher oxidation state. So that is why UO2 acts as an electron acceptor. Whereas SO4, it is also acts as an electron acceptor in case of oxidation reaction. So this SO4 will be accepted as an electron acceptor and converts into sulfide or sulfate. In case of chlorinated hydrocarbons, some chlorinated hydrocarbon can act as an electron donor, but commonly they are considered as an electron acceptors. But benzenes are normally electron donors. Where TNT, what happens is its application is in explosive, and this is also acts as an electron acceptor due to the presence of their nitrile functional group in their aromatic structures. That is why it acts as an electron acceptor. So option A, B, E is electron acceptor, whereas option C, some case of chlorinated hydrocarbons acts as an electron donor, but commonly they are electron acceptor. So the only option D, which is benzene, acts as an electron donor among these five options. So we will move on to the next question. The fundamental requirement of bacterial metabolism do not include option A, carbon source, option B, energy source, option C, oxygen source, option D, electron source, option E, electron sink. So the question is the fundamental requirement of bacterial metabolism do not include option A, carbon source, option B, energy source, option C, oxygen source, option D, electron source, option E, electron sink. So you can post your answers now. And your answers are the fundamental requirement of bacterial metabolism do not include option E, option C. Okay, so let's see the answer. Correct answer is option C, because many bacteria do not need, or even they are armed by oxygen, right? So we call this as anaerobic microorganism. So there are bacteria which do not survive in the oxygen. So these organism we call it as anaerobic microorganism which can survive in the absence of oxygen. That is why the fundamental requirement is not an oxygen source. Whereas all other source they are fundamental requirement. And why it is so? If you see the carbon source, it acts as an building block for the cellular structure as well as for their energy production. If you see the energy source, yes, it obviously helps us to drive the cellular process and for ATP synthesis. And sometimes, for example, glucose acts as the carbon source as well as the energy source for bacterial metabolism. Whereas in electron source, it is mostly useful for carrying out the redox reactions for both energy production and the biosynthesis. Whereas electron sink, what is electron sink? The molecule which accepts the electron, which allows the complete of this redox reaction. So carbon, energy, electron sink, they are the fundamental requirement for all kind of bacterial metabolism. And if you see the oxygen, in case of anaerobic bacteria, we do not see the fundamental requirement is oxygen source. So the oxygen source is the answer, option C. So we will see the next question. The process of synthesizing new cellular material is known as Option A, anabolism. Option B, catabolism. Option C, metabolism. Option D, embolism. Option C, holism. So, the process of new cellular synthesis of new cellular material is known as option A, anabolism. Option B, catabolism. Option C, metabolism. Option D, embolism. Option C, holism. So, what is the answer? You can type your answer, and your answers are the process of synthesizing new cellular material. Option A. Okay, let's see the answer. Yes, it is correct. Option A. And we know that anabolism means it refers to the metabolic process where simpler molecules converting into a complex one, which is building up the cellular materials. So, so what about the other option? Catabolism means it is nothing but the opposite of anabolism. That is breaking down the complex substance to produce energy. Whereas metabolism, it is combination of both catabolism and anabolism for carrying out all kind of chemical reaction. Embolism, it is nothing connected to the bioremediation aspect or the microbial aspect. It is like some blockage in the blood vessels by forming a clot or other material. Okay, so this is no way connected to the macronism and holism. It is a physiological approach which considered whole system. So this is no way these option D and E, they are no way connected to the microbial metabolism, whereas between A, B, C, the correct answer is anabolism, which synthesis new cellular material. We call it as anabolism. So we move on to the next question. Which compound resists the biodegradation in most environment? So, which compound resists biodegradation in most environment? Option A, persistent. Option B, recalcitrant. Option C, xenobiotic. Option D, antibiotic. Option B, E, abiotic. So, among these compounds, which shows the resistant biodegradation in most of the environment? You can start answering the question and post your answers in the chat box. Your answers are which compounds resist the biodegradation in most environment? And your answers are okay. Option A. Okay. Option B. There is no specific example of B. Recalcitrant means normally what? First, we will see the answer. So your answers are like option A and B. Okay. First, we will see the answer. Recalcitrant. And what do you mean by recalcitrant? By definition itself, there is no specific example for B. Okay. These are all like substance that resist the biodegradation. So recalcitrant means they are very slow to break down by the microorganism. Because like recalcitrant, we can say some examples like heavy metals. Some examples, heavy metals and the emerging pollutant, they show some recalcitrant capacity for this biodegradation. So they usually resist the biodegradation. That doesn't mean that we cannot remove the organic pollutant. So in that case, what they will do is uh, they will they will go for some physiochemical treatment or advanced oxidation type of treatment, or sometimes they couple the degradation followed by the advanced oxidation. So these kind of strategy normally scientists will follow to reduce their recalcitrant in nature. So generally, recalcitrant compounds they resist biodegradation, not the degradation. Whereas if you see the persistent, means persistent, the term itself denotes they will stay for long period of time. But that doesn't imply they are resistant to degradation, right? It may stay for long period of time, but if we use an appropriate microorganism, it can be degraded. Whereas in recalcitrant, they resist the biodegradation. We have to go for other way of approach like advanced oxidation or physiochemical approaches. First to reduce the recalcitrant nature, then we can go for biodegradation. Whereas in xenobiotic is nothing but it is a like foreign compound or mainly man-made pollutant that are introduced to the ecosystems. So they are not necessarily need to be resisting the biodegradation. But some xenobiotics are recalcitrant, but not all compounds are all recalcitrant or xenobiotic. An antibiotic, as you said, it is correct. They normally what they do, they kill or inhibit the growth of bacteria. So this is they do not resist the biodegradation. Whereas abiotic is nothing but what does it imply? It is non-living things, anything that is non-living. It can be an environmental condition like temperature, pH, or oxygen level. So it doesn't apply to the concept of degradation. That is why the most appropriate term is recalcitrant, which resist the biodegradation. I hope you got clarity and differences between these A, B, C because persistent means it will stay for long period of time, but it doesn't necessary that it resists the biodegradation. Whereas in xenobiotics, yes, some xenobiotic compounds are recalcitrant, but not all xenobiotics are recalcitrant. You have to understand the difference and you have to read the questions carefully so that we can answer these questions effectively. But you have given a good try, Tuhin and Ashwini. We will move on to the next question, which is mercury is a toxic metal when they are present in the free state. That is, if they are present only in the Hg, mercury state, it cannot easily enter into the cell membrane. But, however, some bacteria, what happens? They methylate these mercury and they form CH3Hg derivatives. So, once it forms these derivatives, now it is very easy to penetrate and they cause more damage to the cells. So, such process falls under which category? Option A, bioremediation. Option B, biostimulation. Option C, bioaugmentation. Option D, activation. Option E, mineralization. I will repeat the question. So, as we know, mercury is the toxic metal, and we have discussed three types of heavy metals. One is essential, non-toxic. Second category is it is essential but toxic at the higher concentration, and third is hazardous and toxic level. So this comes under the third class of metals, which is not toxic. And the one such example is mercury. So what happens here is if it is present in the original state like mercury, it cannot enter into the cell membrane. But once this mercury has been methylated and forms CH3 mercury complex, then now it became very easy to access this membrane. So what do we call this process? Bioremediation or biostimulation or bioaugmentation or it is activation or mineralization. You can read the question carefully and you can answer the question. And your answers are Option C, option B. Okay, so let's see the answer. Option B and C. The correct answer is option D, activation. And why it is so? First, you have to understand what is activation. So it is a kind of biotransformation which converts a pollutant to more toxic product. So by definition, you understand that a normal pollutant is converting into more toxic form. How? If you take this the above example, so normally it exists as an mercury. But now what happens due to the bacteria which methylates this mercury to methyl mercury. Now this less harmful substance is converted into more biologically active and toxic form, right? So if this methyl mercury, high amount of methyl mercury enters into the uh, cells, bacterial cells, they have said that it causes more damage. That is, they became more toxic to the cells. So the bacteria which methylated it, it got damaged and it got killed or it got inhibited. So it this we call it as an activation. So this is why this scenario is best suited for activation. And why not the other options? First is bioremediation. Bioremediation is a like general term where it uses microorganism to clean up the environmental pollution. But here what happens? It causes more toxic to the environment by methylating it. So it doesn't suit for the above situation. Whereas in biostimulation means we will be giving any nutrient or we will be changing any environmental condition to promote biodegradation. But in this case, we are not giving any ingredient or environmental condition to the bacteria to degrade the mercury, right? So it is not a biostimulation. Whereas in bioaugmentation, we will be introducing a specific microorganism to enhance the degradation of pollutant. But here what happens? We have introduced one bacteria, but instead of remediating it, it causes more damage to the cells and causes toxic effect to the environment, right? So bioaugmentation will also not be a suitable option for it. Whereas mineralization means it will be converting the mercury into mineralized compounds like CO2 and H2O. And we all know that mercury it can be changed from one form to the another form and it cannot be mineralized. So the correct option for the above scenario is activation. I hope you got the idea of what is activation and how it is used in the following scenario. So we will move on to the next question, which maintains the bulk precursor pool and allows complete oxidation of the substrate. Option A, mineralization. Option B, assimilation. Option C, oxidative metabolism. Option D, central metabolism. Option E, secondary metabolism. So among this, which maintains the bulk precursor pool and allows complete oxidation of the substrate. You can post your answers. Which maintains the bulk precursor pool and allows complete oxidation of the substrate. You can post your answers. Okay, Thuyan, if you got clarity, it is fine. So the options are D and E. Okay, let's see the answer now. The correct answer is option D, which is central metabolism. So first, why it is central metabolism, and you have to understand what is central metabolism. It refers to the core metabolic pathway. What kind of metabolic pathway we are concerned on? It can include glycolysis, TCA, or pentose phosphate pathway. So these central metabolism, they are the fundamental for the cell survival and growth. And these pathway, what happens? They convert the simple substrates like sugars into metabolic precursors and energy, like ATP and other reduced agents, which are used for both building cell components as well as breakdown of molecules. So this central metabolism can do anabolism as well as the catabolism. And by definition, we understand that metabolism is nothing but it is catabolism and anabolism, right? So for efficient functioning of the central metabolism, we have to ensure that the cell has readily available supply of precursor for biosynthesis of all other cellular components, which oxidizes the substrates for maximum energy yield. So in order to maintain bulk precursor full and allow for complete oxidation of substrate, central metabolism is the right answer. And why not the other one? If you see the mineralization, it is a decomposition process. What happens here? The organic matter is converting into inorganic substrates. So it doesn't maintain the bulk precursor pool. Whereas in assimilation, it is nothing but it is incorporating of nutrient from the environment to the organism. So it is no way connected to oxidation of the substrate. So if you see the oxidative metabolism in case of option C, so while central metabolism, so in central metabolism itself, we are including the oxidative metabolism. So central metabolism includes the oxidative. So it refers to it uses oxygen as an electron acceptor to produce ATP rather than the bulk precursor pool. Whereas in secondary metabolism, here what happens? Production of specialized metabolites. So they are not directly essential for their growth. So among these options, oxidative metabolism suits for maintaining whole bulk precursor pool and allows complete oxidation of substrate. So we will move on to the next question. During biodegradation of xenobiotic substance, having which of these triad will make it degrade the quickest? So here the question is you have to degrade a xenobiotic substance. So what triad or character will make it degrade the quickest? Option A being structurally similar to biomolecules. Option B having a linear carbon chain. Option C having unsaturated carbon bonds. Option D being aromatic. Option E being unsubstituted. So among this, during biodegradation of xenobiotic substance, having which of these triad will make it degrade the quickest? You can post your answers. So you can post your answers. And your answers are option A. Okay, we will see the answer now. Yes, it is correct. Being structurally similar to the biomolecules. Why it is so? Because if the xenobiotic compounds are structurally similar to the biomolecules can be any carbohydrates, proteins, or similar compounds. If they are more likely to be similar, they'll be co-targeted by the enzymes that are capable of degrading these biomolecules. So we can easily degrade these compounds if they are similar to the biomolecules. And why not the other options? So if they are having a linear carbon chain, degrading the linear carbon chain is also very easier when compared to the branched structure. But if overall structure, but here you have to consider the overall structure. If it is very similar to the biomolecules, it will be more easier than the linear carbon chain. So the linear carbon chain can also contain some complex molecule with unusual functional group. So in that case, the difficult to degradation may be higher. That is why linear carbon chain is difficult to degrade compared to the structurally similar to biomolecules. And if you see the unsaturated carbon bonds, which is nothing but unsaturated means they have double bond or triple bond in their structure. So what happens if they have double and triple bonds? It is obviously going to add stability to the molecule. Once it has higher stability means obviously it is going to show some resistance to biodegradation. That is why it showed it is very difficult to degrade if they have unsaturated carbon bonds. And being aromatic means aromatic is nothing but a ring benzene ring with ring shaped structure which show stable ring structure, which is obviously going to resist the biodegradation compared to the structurally similar to biomolecules. And being unsubstituted means presence of some specialized substitute can also influence the molecule susceptibility for microbial attack. So among these statement, being structurally similar to biomolecule is very quicker to degrade compared to other options. So we will now move on to the next question. Antibiotic resistance is associated with frivolous consumption and improper disposal of antibiotic. However, antibiotic resistant genes have been detected in pristine habitat like Antarctica. How this resistance has been occurred? Or they could have arisen because option A, all possible genes exist everywhere. Option B, antibiotic have existed in nature before humans. Option C, antibiotic resistant genes have alternate functions. Option D, antibiotics have a simpler structure. Option E, antibiotics are structurally similar to common substances. So among this, which suits best for the above question? You can post your answers. So what is the answer? Option B. Okay. Now we will see the answer. Yes, it is correct. Antibiotics have existed in the nature before humans. This we have discussed early in the lecture. What happens? Most of the antibiotics they originate from the natural source and they have existed in the environment for the long period of time. Which what happens? The microorganisms evolve their resistant genes against it. In fact, newer antibiotics are synthesized deliberately by introducing structural elements which are uncommon that makes them recalcitrant. But most of the antibiotics they exist in the nature before humans. That's why they develop antibiotic resistant genes that are normally present in their habitat even in Antarctica regions. So what about the other options? If you see the option A, while the genetic diversity is vast, it is unlikely that every possible gene exists in every location. So this is not by the reason for the above scenario. And if you see the option C, some resistant genes might have secondary functions also, but primary role is to confer the resistance to the antimicrobial compounds. Whereas in option D, the complexity is nowhere related or irrelevant to their existence in the nature, right? So this is irrelevant to the this situation. If you see the option E, some structurally similarity might exist between the certain antibiotic and natural antimicrobial compounds, but it need not to be the primary reason for widespread presence of the antibacterial resistant gene in nature, and that too in places like Antarctica. So the correct option is antibiotic have existed in nature before humans that could have conferred the antibiotic resistant genes even in the pristine habitat like Antarctica. So the question 9, which of these nutrient biosynthetic product pair is correct? Option A, SB amino acid. Option B, Mg, ATP and DNA. Option C, PO coenzyme. Options D, calcium stabilizing cell wall. Option E, lanthanum stabilizing ribosome. So among these, which nutrient and biosynthetic products pairs are correctly matched? You can post your answers. Option D, okay. Option B, okay. Option D and option B. Okay. Here you have to observe for nutrient and their product. That is like function, biosynthetic product. So the correct answer is option D, calcium. Why it is so? Because lipopolysaccharide is believed to be stabilized by the presence of calcium ion. And why not the other? If you see the first option, antimony, which is SB, it is not directly involved in the amino acid synthesis. So it is not connected to the amino acid synthesis. So it is obviously false. Whereas in magnesium, it is a metal which acts as a cofactor that binds to the ATP. So magnesium, what happens? It binds to the ATP and it catalyzes the process. So it does not produce the ATP or DNA. Magnesium acts as a cofactor and it binds to the ATP rather than the product. That is why option B is false. And if you see the PO, which is polonium, it is a radioactive element. So it is not an essential nutrient. So it is not obviously going to act as an coenzyme. Whereas in option D, lanthanum, it is there is no scientific evidence that it helps in stabilizing the ribosomes. So the correct option is calcium, which helps in stabilizing the lipopolysaccharide, which is the cell wall of the microorganism. So the last question for the today's session is a wastewater treatment plant with activated sludge system receives the water which contains both organic matter content and the toluene. The level of toluene was determined to be non-toxic to the bacteria. So they clearly mentioned that the activated sludge, they are not toxic to the bacteria. That is, the toluene is non-toxic to the bacteria that are present in the activated sludge system. So after few days, the organic matter was found to have partially degraded organic matter, but the toluene content largely remains the same. So this is most likely to be occurred because of what reason? So what they observed is, they observed after few days, they observed there is a decrease in the organic matter contaminant. So partially degradation was observed in the organic matter, but the toluene was present as it is. Why it is so? Option A, due to lack of phosphorus, or it is due to lack of nitrogen, or it is due to presence of heavy metals, or due to presence of high organic matter in the treatment plant, or it is due to toluene is not biodegradable. So among this statement, which is true? Option B and D. Option B and D. Or okay, or okay. Now only you are answering. Okay. Option E, because toluene is non-biodegradable. Okay. Any other answer? Any other answer? What might be the reason for this observation? You can read out the question carefully and you can answer it. Here, what happens here is they are treating with the bacteria, and they have already said that bacteria is not toxic to the toluene also. But what they observe is they observe partially degraded organic matter, but not with the toluene. Why it is so? Question if not visible one more time. Okay, okay, okay. You can read the question. Oh, option D. Okay, Tuhin. Okay, let's see the answer. Yes, the correct answer is option D. Why it is so? Because here they have mentioned that there is high organic matter content on the toluene. So clearly it mentions that high amount of organic matter is present. So what happens? The microorganism obviously they are going to degrade first the organic matter instead of difficulty to degrade the aromatic compound. So if you compare organic matter and the benzene compound, which is aromatic compound like toluene, which is more easily assimilated a compound, yes, obviously it is an organic matter. And in question they have clearly mentioned there is an higher amount of organic matter. So we we observe that high organic matter has been partially degraded. So if you keep it for longer incubation time, where the exhaustion of organic material happens, then only it initiates the toluene degradation. But in question itself, they have observed that only after a few days, there are still some organic matter is present in the wastewater. So the microorganism will first fully consume this organic matter, then only it will start to degrade the toluene. That's why we could observe there is no change in the toluene content. And why not the other options? If you see the option A, as we know that phosphorus is an essential nutrient, but its lack would primarily affect the degradation of organic matter only, not necessarily biodegradable toluene. So here, if there is a lack of phosphorus, we could have observed there is lack of degradation of organic matter also. Similar to the lack of nitrogen. Nitrogen is used for bacterial metabolism. So its absence obviously do not prevent the degradation of toluene. Whereas if you see the presence of heavy metals, so heavy metals obviously it is going to inhibit the bacterial activity. So it states that toluene is not toxic to the bacteria. So it is not going to be a primary reason, and toluene is not biodegradable. So it is not like that, right? Toluene is also biodegradable, but here the due to the presence of higher organic matter, which is more readily available or accessible as a food source for the bacteria. So that's why they are allowing them to degrade while leaving the non-biodegradable toluene. So if we keep it for more longer time, obviously toluene is going to be degraded. But since there are more organic content, it will be taking more amount of time. So that's why option D is more appropriate for this scenario. So So that's all for the today's session. Thank you for patiently attending this lecture. So this is the 11th interactive session. So the next is going to be the last interactive session for this NPTEL course, Environmental Biotechnology. And one kind request is that you can, for the next session, you can attend it or you can come early because the next section we are only going to deal with the MCQs. So there will you can expect at least 25 to 30 MCQs in the last week 12 live interaction session. So I request you to attend it early. So try to attend at 5 o'clock so we can so that you can solve as many as MCQs as possible because the last week 12 live interaction session of my lecture will be of mostly MCQs. So we will be sharing around 25 to 30 MCQs. So you will be prepared for your final exam. So you try to attend it at 5 o'clock or try to join between 5 also. So it will be very useful for you. So that's all for the today's class. If you have any doubts, you're always welcome to put your query in the above email, or you can put your queries in the discussion forum. You can revisit these videos in the YouTube also, or in the recording sheet of NPTEL Environmental Biotechnology course. So for today's class, that's all from my side. But for the next week, week 12 class, try to attend it early because we are going to discuss only the MCQs, sample MCQs. So there will be no lectures. So try to attend it and get maximum possible. So thank you for attending the course. Now you can leave the session. Thank you.